Okay, so next up is Parul Sharma from the University of Liverpool. Parul is a postdoc.
Yeah, so hello everyone, can you hear me? So thank you, Paul, for my introduction. So today I'm going to talk about animal model, and here I'm talking about comparison of Omicron and Delta variant in mouse model. So before starting, I would like to say thanks to my team because obviously this is team efforts, and without them it wouldn't be possible. Especially Adam and Ali, who is equally involved in this work, and also my PI James Stewart. And Anya Kipper, she has done tremendous job on pathology. So everyone know about COVID-19. It is an infectious RNA virus, and it is called COVID-19. SARS-CoV-2 is like positive single-strand virus, and it has several genetic mutation. And it is still a big threat for human beings because it caused lots of economic loss, and 6.9 million people lost their life. So this is just brief outline about variant of concern on the basis of their origin and their mutation. For instance, alpha, it has mutation N501Y and P681H. It is originated in United Kingdom 2020. And same beta, it is originated in South Africa, May 2020. And gamma in November 2020. And then Delta, that is originated in India, October 2020, and it is highly transmissible, and it has lots of antigenicity changes. After that, Omicron came in November 2021, and due to 30 mutation in spike protein, it is now dominant strain of SARS-CoV-2. So this is a brief outline of Omicron sublineage, and here you can see BA1, BA2, 3, 4, and 5. So these variant also going on, undergoing on different mutation. And for example, for BA4 and 5 is originated BQ1 and BQ1.1. And most recently, BA2, A.6, that is also known as Pyrola strain. So for my study, we use BA1, BA2, 5, and Delta in mice model. So these viruses we got from a Winde Barclay lab, and we sequenced them to confirm any additional changes in the genetic code. And this is brief outline of my in vivo experiment. So we use 6 to 8 week old K18 transgenic mice. And these mice have S2 receptors in their lungs and respiratory tract. So we infected mice with 10 to 3 or 10 to 4 PFU of virus in 50 microliter of PBS. So here in this table, you can see. So yeah, in this table, you can see we infect 10 to 4 PFU of BA1, 2, and 5. And for Delta, we use 10 to 3 PFU. And we also put one group just for uninfected control. So after infection, we weigh them every day to see any possible clinical sign or weight loss. And on the basis of their clinical sign, we cull them either day 6 or day 7. On the cull day, we harvested lung tissue, nasal tissue, and brain for further investigation. So this is brief outline about downstream processing. So we extracted RNA from lung, brain, and nasal tissue. And we did one-step RT-qPCR to identify nucleocapsid N gene. And we also standardized that with against 18S, that is housekeeping gene. And we sent left lung and brain after fixation to University of Zurich for further pathology investigation for SARS-CoV-2 and antigen. So here is my result. So in this graph, you can see this is weight loss curve. So x-axis is showing day post-infection, and y-axis is showing percentage of original body weight. So green line represent Delta. So in Delta, you can see the mice, they lost weight after 2 days, and it's like sharp loss of weight in 5 and 6 days, and it is unrecovered. So we had to cull these mice on day 6. But for BA1 and B2, it's not very significant weight loss. Interestingly, in BA5, we found a weight loss on day 4, but unlikely Delta, they recovered after that. So that is interesting finding, and we can say like Delta caused more weight loss compared to BA1, BA2. So after that, we analyzed tissue viral load. So by qPCR, So in first graph, you can see this is nasal viral load. So here we also found similar thing. In Delta, you can see high viral load as compared to BA1, BA2, and 5. In lung also, we found high viral load for Delta. For BA5 and BA2, BA1, we also got a little bit more, but I mean, it's not that much. So on the basis of these two graphs, we can say in lung, there is like more viral load as compared to nasal tissue. And it is also like day 7 post-infection. So maybe at that time, virus is cleared from the system. Interestingly, when we done plaque acid from day 1 post-infection lung lobes, so here you can see BA5 has more viral load as compared to Delta. So maybe Omicron, they replicate at early post-infection, and after they declare from the system. That's why you can see more virus load in day 1. So this is lung histopathology slides. So this is also telling similar story. In BA1, BA2, BA5, it's like a small consolidation and unaltered parenchyma. But in Delta, you can see the brown patches, that is like virus protein. So Delta has high viral load and uneven lung parenchyma that increase cellularity. So we also interested to see like if virus can enter any other tissue. So on the basis of neurological symptom like loss of smell and loss of taste, we thought maybe we could see the brain tissue. And for that we done PCR on brain tissue. And here also we find similar thing. Delta has high viral load as compared to BA1 and BA2. Interestingly, BA5, you can see some viral load here. And pathology also telling similar story. Delta has high, like wild spread neural infection and mild non-supporting encephalitis. In BA5, we found some neuronal changes and but there is no inflammatory response. So on the basis of this, we can say BA5 and Delta can cross the brain barrier. So on the basis of these findings, we can conclude that Omicron variant causes less clinical sign. For example, weight loss. BA5 is slightly more pathogenic as compared to other Omicron. And qPCR data show like high viral load in lung. Also, Delta and BA5 can cross brain barrier. So finally, I would like to say thanks to UK G2B Consortium for providing funding for this work. And also I'm grateful for technical staff of University of Zurich to perform pathology and biomedical science service unit, University of Liverpool, for providing animal facility, UKICN to giving me this opportunity for presenting my talk, and University of Ljubljana to hosting us, and all of you for listening. Thank you.
Any questions for Cheryl? I'll start actually. The striking difference in neurotropism, do we understand the molecular basis for that or have hypotheses for it? What's the difference?
What do you mean by?
Well, why does Delta spread so much to the brain compared to the others? Is it just a factor of virus load, or can we take a guess at a specific sequence difference that makes the
Actually, we haven't done any sequencing, but maybe we could do that. And also maybe there's like different, entry, like Omicron is used endosomal pathway, and maybe Delta is using templars. So that also make difference.
Because my thought process is, as some of you know, I'm really a flu virologist, and in the flu system, the thing that makes the big difference for neurotropism in avian species and in mice is the cleavability of the viral glycoprotein.
Yeah.
I mean, do you see differences in the furin cleavage sites in your?
We haven't done any in vivo work, in vitro work for this thing. Yeah. So.
Any more questions for Cheryl? Misha.
Hi.
Yeah, now it's working. Very nice work. I was just wondering, did you maybe look at the earlier lineages too? Or just starting from Delta and Omicron? Did you look at the Alpha and in the same experiment settings?
Yeah, we have done actually Delta and like Wuhan strain in early days, and also we thought, we saw the same findings. So like Omicron is high in like early days, yeah.
So Omicron is similar to the early days, or Delta is similar to the early days?
Omicron is like early days and other strain like alpha or like they are like later. Yeah.
Okay, thanks.
Thank you.
Anyone else? Oh, sorry.
That's fascinating data, and the mouse model is obviously a very useful tool. From memory, the BA5 data that were generated in hamsters
Yeah.
does show a difference, but I think the BA5 in hamsters looks not as bad as Delta, but not as poor as in the mouse. And I wondered if you've got a comment about that.
Yeah, actually we have done BA5 on hamster.
Yes.
Yeah, and we also got like some weight loss like after 3 or 4 days. Yeah, but like it's I mean after 4 or 5 days, just hamster recovered for that thing. So I mean it is not like Wuhan strain or Delta strain, but still it's caused some weight loss. Yeah, and viral load.
Okay, well, thank you, Cheryl, for a really interesting tool. Beautiful data.